through the Q&A feature. Okay, I'm going to hand it over to you now, Tyler.
Thank you so much. Well, good afternoon. My name is Tyler Trent. I am the SPSE Application Specialist. I specifically work with the Automate Q40. So, my presentation today is entitled QuECHERS, a practical approach with implications beyond the original design. In today's presentation, we're going to be covering topics of what is QuECHERS, a little bit of troubleshooting of QuECHERS, the Automate Q40, which was designed by Teledyne Tekmar, and then two experiments that we went through. So one is an apple juice validation with a partner laboratory, and then a validation of the EN 1566 2008 method for QuECHERS also. So what is QuECHERS? QuECHERS is a technique to extract pesticide residues from plant materials. Many people have many different ways of saying QuECHERS. Well, it is pronounced as QuECHERS, as you can see on the screen here. It is a portmanteau word, basically meaning quick, easy, cheap, effective, rugged, and safe. While the first method of QuECHERS was actually developed back in 2003 and presented at the EPRW overseas, it was developed by Michelangelo Anastasius, Stephen Lahote, and others within their group. It is designed to extract pesticide residues in agriculture commodities such as fruit and vegetables, as you can see off to the right. But since its implication, it has been expanded to many different matrices. So QuECHERS has been applied to grains, processed foods, honey, nuts, animal tissues, wine and beer, water, compost, soil, tobacco, dietary supplements, and many more are being added to this list as we speak. So over the 10 years QuECHERS has been around, QuECHERS has gained a lot of popularity within commercial food testing laboratories, food producers, manufacturers, regulatory agencies such as the USDA, FDA, even overseas at the European Food Safety Administration, the EPA, and also within public and private auditing companies. These auditing companies must know the method in and out so they can provide a complete and clear audit for the company. So, more introduction to QuECHERS is, QuECHERS offers a very attractive alternative to past sample extraction techniques. So, with this is fewer steps, less solvent consumed, no chlorinated solvents such as the Luke method, and it's also valid for a wide range of pesticides, over 800 types and still growing. Also, your consumable list is going to be cut down also. You need less glassware, no large volumes of solvents, smaller sample size, and easier disposal since it's not chlorinated waste. As QuECHERS has been developed over the past 10 years, it continues to undergo modifications from the original method. It first started off as the unbuffered method, which basically included magnesium sulfate, sodium chloride, and acetonitrile to do the extraction. But as time has went on, we go through changes, and so does methods. The QuECHERS method has now been developed into the AOAC and the EN method. With the AOAC method, they use a 1% acetic acid and acetonitrile and sodium acetate buffers in the salts. This helps protect base-sensitive compounds and better quality of recoveries for pH-sensitive compounds also. The EN 15662.2008 includes a different type of buffer. It includes a citrate buffer instead of an acetate buffer. This basically will help protect against base-sensitive compounds. Then also towards the end of the method, they will preserve it with formic acid. So there's still an acid step, but it's at the very end for the preservation of the compounds. Right here is basically a QuECHERS method flowchart, going from the original unbuffered QuECHERS method to the AOAC to the EN. As you can see here, the sample ratios are about the same. Sample ratio and solvent ratios are always the same. We're working with a 1-to-1 ratio, so 10 grams to 10 mils for the unbuffered. AOAC, you can see you're working with a 15 gram to 15 mil, so 1-to-1 ratio. The EN also employs the 1-to-1 ratio of 10 to 10. Where things get different between the two methods are the salts and buffers that are used. As you can see here with the unbuffered, it uses magnesium sulfate and sodium chloride. But when we go over here to the AOAC, you use magnesium sulfate and then acetate buffers. Then as we go over to the EN, it uses magnesium sulfate, sodium chloride also, but it uses citrate buffers instead. And there's very minimum differences between the methods. One method might call for a 5-minute centrifuge. The other method might call for a 1-minute centrifuge. The cleanup steps are approximately the same, but they use different ratios also. 6 mils for the unbuffered and the EN, then 8 mils for the AOAC. But the PSA, mag sulfate, and the other cleanup materials are adjusted for that extra 2 mils of solvent. And then the other glaring difference between the methods is the EN will preserve with 5% formic acid at the end before it gets sent off to be analyzed. So what does QuECHERS technically involve? It involves homogenization of a commodity, addition of an extraction solvent, addition of spikes in IS if needed, addition of salts and buffers, sample shaking and vortexing, centrifuging, decanting, dispersive solid phase extraction, also known as DSPE cleanup, preparation of the final extract, and then analysis by GC, LC with many types of different detectors at the back end, but typically we'll see a GC MS-MS or an LC MS-MS. So what we're going to do now is we're going to walk through the method itself, going through some steps and showing what is optimal and why they chose to do it. So the sampling homogenization step must be done. It increases the surface area for the extraction, and it also ensures you get a representative sample from the samples that are submitted. As I said back before in the flow charts, a 10 to 15 gram sample is extracted. That is with samples that have moisture contents above 70%. Once you start switching over to drier samples, such as flour, dry fruits, spices, and many more, you need anywhere from 2.5 to 5 grams, all depending on your detection limits that you're trying to hit. Then also with this step, when you're looking at moisture contents below 70, you're going to need to add some water to hydrate it back up to the optimal moisture content range for the QuECHERS method. So the first step is basically the liquid solid extraction. Acetonitrile is the extraction solvent of choice. It is designed to remove the compounds from the sample matrix. Solvent choice is very important to reduce co-extracting analytes. As we read through literature over the past 10 years that QuECHERS has been around, we have seen people use ethyl acetate or acidified methanol. With the acidified methanol could be used for such as highly polar compounds such as the QuECHERS method, the QUPPE method that was just recently released. Then after the acetonitrile, the extraction solvent is added, then you're going to add the addition of your salts and buffers. So the magnesium sulfate assists in the partitioning, the sodium chloride reduces the polar interferences, and then as I said before, the buffers, depending on which ones you're using, will prevent compound loss of pH-sensitive compounds. So the liquid-solid extraction continues. The solvents and salts are added to the sample tube containing the commodity. It is shaken vigorously, and the sample is centrifuged. As you can see down here, we're going through the steps. So the sample is shaken, and it is centrifuged for either 1 to 5 minutes, depending on the method, and then you'll get your product looking like this. At the bottom of the right-hand picture, you'll see the excess salts, the water layer, and then you'll see the food plug on top Then on top of that is your acetonitrile layer, your supernatant, which contains all your compounds that you are extracting off of your sample. The next step as we progress through the method is a dispersive solid SPE step. This step can either be performed or not. We can actually take the liquid on top of our liquid solid extraction if it's a easy sample such as apple, and we can just dilute and shoot into our LC or blow it down and shoot it into our GC. But if you do need a cleanup, we will go through the dispersive solid phase step. Basically, we will transfer an aliquot, 6 to 8 mils, if you're using the full-size tubes, or 1 to 2 mils, depending on if you're using the micro. The aliquot of the supernatant is decanted off into a centrifuge tube containing the DSPE for the cleanup, and is shaken for a minute. There are a lot of different types of combinations of cleanup out there. As I listed, just the 4 primaries that can be used. The magnesium sulfate is typically always present because it removes excess water from the organic phase. Typically, the PSA, primary secondary amine, is always present also. It will remove the sugars, fatty acids, organic acids, and lipids. But sometimes this will grab a hold of certain compounds, certain groups off your pesticides, and will retain them. So you have to be very careful on the chemistry there. Then also C18. C18 can be added or removed into these cleanups. C18 will remove long-chain fatty compounds and nonpolar interferences. Typically, a C18 will be used when you're looking at a grain sample or something with high fats, maybe like an avocado. GCB, also known as graphitized carbon black, will remove pigments, polyphenols, and planar compounds. So when you use GCB, you have to be very careful because it will attach itself to your planar compounds. And I'll show you, there's a tip down the road later on in this presentation of how to get those compounds released back up into the supernatant instead of being held onto the GCB. So right here is just basically a picture depicting the dispersive cleanup step. Over here in the top left, you see that we're transferring the liquid over from our liquid solid extraction tube over to our dispersive solid phase extraction. We'll shake it, spin it down. Then after we spin it down, we'll see different types of things. Over here, down the bottom part, is 2 different types of commodities. One's a strawberry and one's a spinach. And they are taken through 2 different types of cleanups. One cleanup will have PSA, magnesium sulfate, and GCB. The other one will just be magnesium sulfate and PSA. As you can see here, the strawberry really does not need the GCB, which could cause some issues down the road on the analytical side because it's highly selective, so it might have grabbed a few compounds out of the supernatant that you are actually analyzing for. So for the strawberry, you may not need GCB, but when you come over here to the right and start looking at the spinach sample is dark green, very rich in chlorophyll. Chlorophyll is also going to be an interference for you. You need to help minimize that. So we'll add in some GCB to go ahead and grab onto that chlorophyll. As you can see here, 2B on the right-hand side is basically magnesium sulfate and PSA. It does not really remove much pigment. But as soon as we place in graphitized carbon black, you'll see the removal of the pigment in tube C, which should give us a cleaner extract to use. So after you've been through the liquid solid extraction, the DSPE cleanup step, now your sample is ready. Now you have your final sample. So basically you'll transfer the organic layer off the top of the DSPE bed into a 2 mil GC or LC sample vial. You can either do a dilution or a concentration step. Typically, you're looking at a range of 2 to 10x concentration or dilution. The extract, the amount of dilution may be varied because of different types of instrumentation. So as you see here, we have an LC triple quad, and a GC single. These samples can be run on GC triples, GC ECDs, and so on. Those might have to be blown down in solvent exchange because of acetonitrile may not be as great for a GC inlet or the source. So as we move through this presentation, we're going to start talking about a little bit of troubleshooting that can come along with QuECHERS. So the first thing we ran across, one of our customers brought us an issue at hand, is a emulsion layer. So as we all know, emulsion layer is a mixture of 2 or more miscible solvent layers. The sample over here on the right is a soy grain, so it's very high in protein. And no matter what we did, we kept on getting this nice thick emulsion layer on top. Well, we tried to cut down the smaller sample amounts to cut down on the emulsion layer. That worked a little bit. But one thing that we realized is when I did some research on veterinary residues is that they swapped out magnesium sulfate for sodium sulfate as the extraction salt, and it creates a less exothermic reaction, and we'll get a very small emulsion layer, but we still actually have our supernatant on top that we can actually transfer over. This is still in works. We still need to go through a full validation to make sure that it is correct, but this is one of the experiments that we've actually started working with and seeing how different things of swapping out different salts will play within the QuECHERS extraction. Well, what if you're having recovery issues? There's a few ways to get around recovery issues that you can do. One, use a matrix max calibration standard. Go out and find a clean organic sample of the same commodity and run it as your matrix match. Or the use of internal standards. Internal standards have changed every time. Typically, you see a lot of people use TPP, triphenyl phosphate. But now, with some of the newer methods, people are starting to use deuterated. So there's 2 things that they can do there. They can use a TPP or actually use a deuterated and correct for recovery loss. Or say if you have poor extraction efficiency. As I said before, we got to make sure our sample is hydrated. So we're looking from that 70 and above for efficient recoveries. Or we didn't choose to correct buffers, which is required for pH-sensitive compounds. Pigmentation and matrix issues are other things that we come across. So if you have dark or high chlorophyll samples, you might want to use GCB to remove pigmentation and the chlorophyll, as I discussed before. This also, once you start using GCB, it might have some reduction of planar compounds. To get some of those planar compounds back from the DSPE bed, you might want to add some toluene to help increase your recoveries of your planars. As I said before, GCB is very highly selective. And with a lot of customers I have talked to and discussed this with, if they use GCB, they use the European version of the GCB because less GCB is better because it's so highly selective that it could go and actually take out other things that you're needed to be looking for. Or say if you have high fat samples such as avocados and grains and commodities along those lines, you might want to use a C18, a long chain fatty compounds for this one, and nonpolar interferences. One thing that people have been doing is using analyte protectants. Analyte protectants are basically used when they are running on a GC, due to the strong interactions with active sites in the GC, such as in the inlet, column, or ion source. Sometimes you'll see degradation, absorption of co-injected analytes. You'll start seeing tailing of peaks to make it very not easy to quantitate. So basically, we will add in analyte protectants, which will give us sharper peaks, less tailing, and it will make our GCs more rugged. So we can get away with more injections before changing out consumables on that. This is a slide that I got from a presentation given by Agilent. Shows QuECHERS, basically, a QuECHERS extraction being ran on a GC with and without analyte protectants. As you can see on the left, with analyte protectants, you get a very nice, sharp Gaussian peak that you can integrate very easily. But on the right, without analyte protectants, you start seeing shouldering, you start seeing tailing, and it's just not pretty, which might make it a little bit more difficult to integrate. So using analyte protectants will help you in most cases. What we see is there are a few challenges for QuECHERS. It offers a very alternative to past sample extraction techniques. It's dirty, it's quick, it's easy. But one thing that we have seen is increasing number of samples that must be analyzed. And however, it's very labor intensive because you have to weigh out, shake, centrifuge, add solvent, add salt. So it's very labor intensive. Depending on how many samples you're actually running, it could take up to 8 hours for a batch. What we did as a company is we came out with an automated extraction system that's capable of extracting 40 samples. It's called the Automate Q40. It is designed to automate the original unbuffered method, the AOAC 2007.01, the EN 15662. The system is capable of method optimization, and handles 50 and 15 milliliter conical centrifuge tubes. So let's talk about what is automated and what is not automated. Let's start off with the shorter list. So what's not automated is you still have to do the sample homogenization. You still have to weigh your sample out. That's basically it. Then the only thing you have to do is load your sample tray. Loading your sample tray consists of placing your sample that's going to be extracted into the slot, loading in the corresponding DSPE for that extraction, loading in the final extract tube where you will have your pure raw extract, and then loading in your consumables such as your solvents, disposable pipette tips, and salts. So now let's move over to what is automated. We have the addition of the extraction solvent is automated. We can add up to 3 types of solvents. Addition of spikes and standards. We can add up to 3 spikes or 2 spikes with 1 preservative, depending on if you're going to run the EN method or the AOAC method. Addition of extraction salts and buffers. Basically, it's a pre-mixed tube that contains your salts and buffers. We're able to shake the sample. We're able to vortex the sample. We're able to centrifuge. We're able to transfer or decant the supernatant through the tubes. We're able to do the dispersive solid phase extraction cleanup. We can do preparation of the final extract, preservation, and then we have something called vial vision, which will basically ensure accuracy of the sample. So automation model. So some of the components that we have here is we have sample trays. It's a 40 position sample. Each tray holds 5 samples. Those are able to be removed from the system. It is a XYZ robotic. It has a vial gripper, vial shuttle, capping and decapping station, pumper assembly for the extraction solvents, as I mentioned before, pumper assembly for the spikes and preservatives, solid addition module, vortexing station, shaking, centrifuging, liquid detection, pipetting, and then a fume hood if you need it. So right here is the overall design of the unit. Everything is, your whole bench is basically thrown inside of the Automate Q40. What I'm going to do right now is actually show you a video of the Automate Q40 in action, going through each sample step, going through each station, and showing you how it is technically used. So basically this is just an overall, so we'll take you from your homogenized sample to your final extract. It's organized with samples, DSPE, and final extract. So we'll add our precision delivery of our solvents. We'll vortex it before the salts are added, if need to be. Then we'll have an addition of salts. your extraction salts. We'll take it over and shake it. After shaking is done, we will centrifuge it, which will separate the organic layers from the commodity. Basically, vial vision identifies the organic layer. Pipetting. We'll move and transfer the aliquots from the extraction tube to the DSP to the final extract. The DSP cleanup stations that are used also are shaken again, centrifuging to spin down the media, vial vision, and then transferring over to the final extract tube. Then after it's all done, it'll prepare the samples, it's capped, and it's returned to the rack. So that is a short video of showing you all the stations of the Automate Q40. One great thing about the Automate Q40 is the user interface. It's very simplistic. Basically, we'll have 4 buttons, your diagnostics, which will have all your accuracy tools and alignment tools inside. Configuration will allow you to name your solvents and standards. Methods is where you develop your QuECHERS method at, and schedule is where you will go through and schedule it. Just to show you quickly what it is like to build a method. So if you click on Method, New Method, it will bring you up to this page right here where it will give you 5 choices. It will walk you through the extraction step by step. You click Next. As you can see here, you can name your solvents that you're using. So you can select, I'm going to add in 10 mils of 15 mils of 1% acetic, go through, click Next. How much salt am I adding? Am I adding 1 rotation, which is set to 6.5 grams? One great thing about the salts is you can change the volume of salts anywhere from 3 to 10 grams of salt. You can spin or centrifuge your sample all the way up to 30 minutes, 60 minutes, depending on what you want to do. Vortex time is how long you want to vortex your sample that has the solvent been added to it. Mix time is how long do you want to mix your sample with the salts and solvent added to it. Then we'll come over to the next page, which is basically the DSPE page. We can either bypass that by clicking No, which will allow us to just do the first part of QuECHERS. But if we do need to incorporate a cleanup, we can click Yes, and we can tell it what we need to do. After that is all set, then we'll tell it how much we want to transfer over to the final volume. So within QuECHERS, within the Automate, the simulator, within the TechTouch, you can go through and optimize your method, customization, if you would like to do a modified version of QuECHERS. Limitations are endless, so you can do what you need to do or try what you want to try. So as we move through this presentation, we're going to go through and talk about 2 types of experiments that we did. This experiment was actually presented at North American Chemical Residue last year. So basically, it was a dual lab validation. Store-bought apple juice was used and tested for pesticide residues. We used the automated extraction. We used the AOAC 2007.01 as the extraction method. We had a sub-list of target pesticides. We had normal QC guidelines. We checked at 10, 50, and 100 nanograms per mil. With this multi-lab validation, we worked with Pacific Agricultural Lab. So they basically had an Automate out there, and we had an Automate here. We ran the same validation study and compared our results to see what we were getting. As you can see here, it's just a standard AOAC 2007.01 QuECHERS extraction method that we followed. We did use a step with a cleanup, and we did use an extraction without a cleanup. As you see here, it's the SRM transition list for LC-MS parameters. We basically ran the same compounds. Most of them had the same transitions. Some of them slightly varied. LC conditions, we ran on a HPLC, LC-MS, MS. They ran on a UHPLC. We did a 5 millimolar ammonium formate. They did a 20 millimolar ammonium with 0.2% acetic acid. Basically just an LC-MS chromatogram from us here at Tekmar. And here are the apple juice results. So we looked at two different types of studies. We looked at a apple juice that was sent through a DSPE cleanup step and without a cleanup step. As you can see, looking at all of the values, is that the values did fall within the range that we were looking for, the acceptable range of 70 to 120. And all the RSDs were well below 20% for the cleaned apple juice samples. Then if we look at the unclean apple juice samples, the same criteria was met. All the sample values fell within 70 to 120, and all the percent RSDs were below 20%. So looking at this apple juice study, all pesticides gave outstanding recoveries and showed excellent precision. A combined average recovery across all levels from the 10, 50, and 100 ppb extraction checks from Teledyne Tekmar and the Pacific Agricultural Lab came in at 100.17%. And then we had a combined precision of 6.76%. So with this, the Automate enables a reliable and more reproducible extraction and also allows significant time and labor savings for the extraction lab. Another validation that we did was a validation of implementation of a fully automated QuECHERS solution for the EN 15662-2008. So basically this is the European version of QuECHERS that we went through and validated. We did an automated extraction according to the EN method. We had a target list of 241 pesticides. They followed normal QC guidelines of 10, 20, and 50 nanograms per gram in the results. I just recently presented this work at ASMS and then also over at the European Pesticide Residue Workshop. This work was done in partnership with Simon Hurd and James Barlow, their group over at FARA, so the Food Environmental Research Agency. So they follow the extraction parameters, 10 grams, 10 mils. But one thing that they varied was that when the samples did need cleanup, they would use magnesium sulfate and PSA. But when they were running a sample they knew that did not need a cleanup, they would just use magnesium sulfate to help dry it out to remove excess water from that organic layer. So basically right here, they ran it on a UHPLC MS-MS. As you can see over there is the MRM chromatogram for the 241 pesticides that was looked for in this validation study. So I know some of these compounds might be very hard to read at the very bottom, but basically you see the green line at the top and the purple line at the bottom. Those are our regions that we need to fall within our 70 to 120. As you can see, we did an apple validation. So they basically looked at two levels. They looked at a 0.01 mg per kg level and then a 0.02 milligrams per kilogram level. And as you can see, all the samples did fall within the range, and only two were outliers. Then continue this validation study is they had to do a validation study on apples, oats, raisins, lettuce, and grains. So they had to cover the wide gamut of compounds that were of commodities that they would be seeing to make sure that Automate Q40 is valid. As you can see here, the oats validation, oats are grains, so it gave a little bit more difficulty. They used 5 grams of sample here, then they used 10 mils of water on top of it to hydrate it. As you can see, all the values fell within the 70 to 120, except for a few outliers, as you see down here. But when those values did fail below those limits, they were all reproducible, and they all happened at all 3 different levels. They all failed at the 3 different levels. So it was extraction efficiency or extraction recovery that dictated those. And as we move through, we also did a lattice validation. As you can see, 70 to 120 also. Not as many compounds fell outside the 70 to 120%. But where the kicker is, the results for the automated procedure were well within the criteria set by the EU AQC guidelines. We met all their requirements for a full validation. All pesticides gave outstanding recoveries and showed excellent precision. We had a combined recovery of 106 to 114% across 6 commodities. Then we had a combined average recovery of 4% across 241 compounds across 6 commodities with 3 QC check levels. As we know, the automated Q40 enables reliable and more reproducible extractions and also allows significant time and labor savings for the extractions. And that is basically it for me. If you guys have any questions, please submit them over to the Q&A box, and I'll be happy to answer them.